In this video, I'm going to go through how gene expression can be regulated at the transcriptional level by transcription factors, with a particular focus on the Lac Operon. Now, you need to remember that a gene is a short chunk of DNA that codes for a protein, and that genes are not always expressed, or sometimes we say that they're not always turned on. And if the gene isn't expressed, then the protein that they code for won't be produced. So we're going to talk about transcriptional level control in this video. There are also post-transcriptional and also post-translational level control of gene expression, but we won't go into those today. And just to remind you that transcription is when mRNA is produced from DNA using the enzyme RNA polymerase, and translation is when that mRNA code is then used to produce proteins at the ribosomes. But we are just going to focus on transcriptional level control. So gene expression and therefore protein synthesis can be controlled at a transcriptional level. And the rate of transcription of genes can be altered. So if transcription happens more, then more mRNA is going to be produced, and therefore more of that protein that the gene codes for is going to be produced. And if transcription happens less, then less mRNA will be produced, and again, therefore less of the protein that gene codes for is going to be made. Now, all of this is controlled by transcription factors. Transcription factors are proteins that can bind to DNA to either increase or decrease the rate of transcription. If a transcription factor increases the rate of transcription, we call them activators, and if they decrease the rate of transcription, we call them repressors. It's the shape of the transcription factor that's key to them binding to DNA, and their shape can be altered or changed by the binding of certain molecules. So, for example, lactose, a sugar, is important for the binding of transcription factors in the case of the Lac Operon, which we'll talk about in a few minutes. So we're going to mainly be focusing on transcriptional level control of gene expression in prokaryotes, such as E. coli, so that's the Lac Operon. But it's also important to know that transcription factors control gene expression in eukaryotes too, so eukaryotic organisms such as animals and plants. So in eukaryotes, transcription factors bind to DNA at a site near the start of the gene that they're controlling the expression of. If it's an activated transcription factor, then this will allow RNA polymerase to bind to the DNA to therefore start transcription, to start the production of mRNA. And if it's a repressor transcription factor, this will stop RNA polymerase from binding to DNA, so transcription can't occur, and therefore no mRNA will be produced and no protein will be produced. So that's eukaryotes. Now in prokaryotes, transcription factors bind to chunks of DNA that we call operons. So when you hear the word operon, just think of it as a chunk or a section of DNA. Now, operons are important because they contain structural genes which are labelled A, B and C, and these genes are all transcribed together and they will code for really useful proteins, for example enzymes. The operon will also have a section of DNA which we call control elements, which normally come before the structural genes. Now these control elements include a promoter that's labelled P, and that promoter is important because it is where RNA polymerase can bind to. And the control elements also include an operator, and that operator is where transcription factors bind to. Now the operon may also include a regulatory gene, which is labelled R, and this regulatory gene will code for a transcription factor, so either an activator or a repressor. Okay, so the Lac Operon. Now we're talking about E. coli, which is a bacteria. Now this bacteria can respire using lactose if glucose isn't available. The important thing is that the genes needed by the bacteria to be able to do this, to be able to respire using lactose, found on an operon, and that operon is called the Lac Operon. Now remember, an operon is just a section of DNA, and it's controlling structural genes all transcribed together, as well as the control elements and sometimes a regulatory gene. Now the Lac Operon contains 3 structural genes to allow it to be able to respire using lactose. There's LAC-Z, which codes for an enzyme called beta-galactosidase, and that enzyme breaks down lactose into glucose and galactose. One of the structural genes is called Lac Y. Now this codes for a protein called lactose permease. It's a cell membrane protein, acts as a pump which pumps lactose into the cell. And the final structural gene is called Lac A. Now less is known about this protein and the kind of function of it. We know that it codes for the enzyme beta-galactoside transacetylase. That enzyme transfers the chemical group acetyl to lactose, but how that helps with the breakdown of lactose is not fully understood. So you need to know that there's 3 structural genes, Lac Z, Lac Y, Lac A, but Lac Z and Lac Y are definitely the important ones in terms of the breakdown of lactose to allow the E. coli bacteria to respire using it. The 3 structural genes on the Lac Operon are only transcribed into mRNA when lactose is present. So this is what the Lac Operon looks like. We've got the Lac I, which is the regulatory gene. It codes for a transcription factor called the Lac repressor. We've got the control elements, so the promoter and the operator. And we've got the 3 structural genes, Lac Z, Lac Y and Lac A. When lactose is not present, the Lac repressor, which remember has been formed from the regulatory gene, the Lac I, and that Lac repressor binds to the operator. Now, if the Lac repressor is bound to the operator, it means that RNA polymerase cannot bind to the promoter, and therefore no mRNA is transcribed, and therefore no proteins that are coded for by those 3 structural genes are produced. And that's when lactose is not present. When lactose is present, the lactose actually binds to the Lac repressor. Now this changes the shape of the Lac repressor, and it means it can no longer bind to the DNA of the operator. Now that means the Lac repressor is released from the operator, and therefore RNA polymerase is now able to bind to that promoter region of the control elements of the operon, and transcription can begin. And therefore mRNA for those 3 structural genes are produced, and that means the proteins that those 3 structural genes code for are now produced. And that enables E. coli to now respire using lactose. In this video, I hope you now feel a bit more confident with how gene expression can be controlled at a transcriptional level, which involves transcription factors such as the Lac repressor in the case of the Lac Operon. And if you want more resources, please head to biologyfocus.com.